Brad Macy, President and founder of Hospice Corporation, and I'm really excited today to present Joe Solien, PharmD. Joe is responsible for OnePoint Care's National Clinical Service Program, including staff and partner education, formulary design and development, and also medication utilization reviews. He's the primary author of OnePoint Patient Care's Clinical Symptom Guide, which is currently in its third edition. He began working at OnePoint as a pharmacy student intern in 2004 and became a staff pharmacist after his graduation. And in 2010, he was promoted to clinical pharmacist, where he's continued to serve on the clinical team in a number of roles. Joe earned his PharmD at Midwestern University, Glendale, and is board certified in both psychiatric and geriatric pharmacy. He also earned his BS in biochemistry and molecular biology and a BA in chemistry from the University of Minnesota at Duluth. Joe, welcome, and thank you for being with us today. I'm going to turn it over to you now.
Thank you, Brad, for the introduction, and thank you so much for having me. Just a quick bit about OnePoint Patient Care. It's a national hospice pharmacy services provider, providing dispensing, PBM, and clinical services to about 450 patients nationally, or over about 40,000 patients in terms of daily census. I'm calling you today from Tempe, Arizona. Don't let the jacket fool you. For us in Tempe, when it's 55 degrees, it is considered cold. So we'll jump in. Our objectives today, we'll talk about what is it about a molecule that makes it ideal for transdermal absorption or not. I want to show you absorption studies in kind of the two different types that are out there, how to interpret results from those. And then finally, talk about some of the pushback that we get against this. We have very good scientific reason to avoid most topical compounds. So what's driving it? I think ultimately, as the title said, we're going to have kind of placeholder slides where those are the five things that we should know. But really, the ultimate objective here today is to, if you're at a program that utilizes topical gels, hopefully you learn enough today to go back to the program and advocate to improve or change that practice. So with that, we'll start off with the first of the five things, basics of transdermal absorption. When feasible, and when I say feasible, I'm talking about when a molecule possesses the specific characteristics that we'll talk about in a bit. Transdermal absorption is really advantageous. It avoids the stomach, so we aren't going to see GI distress. It avoids first-pass effects, so we're not worried about that coming out of circulation and having hepatic metabolism before it reaches circulation. It gives really good, steady drug levels. A biggest reason or a biggest advantage in hospice is in folks with dysphagia or where they can't swallow oral dosage forms. One of the big takeaways today is that when one of your patients has dysphagia, these topical gels are not a good alternative. I'll talk about that drugs need to get into circulation, whether that's through the oral route, sublingual route, parenteral route, if a drug's available in a suppository, the rectal route, or even if it's not, the Macy catheter can be used to administer medications rectally. Back to some of these advantages, it's easy, it's non-invasive, it's probably one of the easiest ways to take a medication, and as such, we see with these dosage forms that patients are often more compliant than they would be with others. That all sounds great, but there are significant disadvantages with specifically the compounded topical products. Far and away, the number one problem with these is that most molecules are not suitable for systemic absorption. We think about this, if haloperidol really was absorbed rapidly and effectively through the skin, there would be a lot of money to be made in making that form commercially available. It would usually, if a drug is suitable for systemic absorption through the skin, it already exists in a commercially available form. Other disadvantages, these are compounds. They don't have standard dosing. They didn't go through the different phases of FDA approval to demonstrate safety and efficacy and ultimately to find an effective dose. And then for some of them, specifically with promethazine, there's some kind of tough to look at pictures of adverse drug effects that have happened following administration of these topical compounds. Getting back to the basics of transdermal absorption, you know, the main two functions of the skin are simple. They're to keep us and keep water inside. The skin holds our insides in, as they say. It's also to keep foreign substances out of the body. You could hold a lead fishing weight, for example, and not get lead poisoning. It doesn't always work that way. Where you can hold a tablet in your hand and you're not going to absorb from the tablet. I want to point out the different parts of the skin. The outer layer is the epidermis, and if you look closely here, you can kind of see it's broken down into two parts. I'm going to zoom in on that in 2 slides here. Underneath that is the dermis, and I'm pointing this out because the epidermis is avascular. It doesn't have capillary beds so that drugs could be absorbed into systemic circulation. The dermis does. That's where a drug would need to get in order to get into circulation. Why does that matter? That's how drug can find their receptor. Okay, this is not up for debate in any way. This is just how drugs work. Drugs get into systemic circulation. We don't have receptors just in case we find a drug someday that works at those receptors. They have what's called natural ligands. So, for example, the opioid receptor, all right, we have mu and other opioid receptors. These are there. The endogenous or the natural ligand are our endorphins. The agonist, of course, would be something like morphine, oxycodone, or familiar opioid agonists. Sometimes you even hear it in the term of the drug, like dopamine agonists, like Requip and Mirapex. We also have antagonists, so that's something that's kind of in the diagram here, jamming the lock. It's preventing the normal agonists from binding to and affecting the downstream changes once that's binding. But the take-home point from here is that we need the drug to get into systemic circulation, and if it doesn't, it's just completely not rational to expect the drug to do anything pharmacologically. All right, so I mentioned that back when we were looking at the better picture of the skin, I think, we would look at the epidermis in terms of its 2 main layers. The outside layer is the stratum corneum. It's about 1/10 of a millimeter thick, representing about 7% of the total thickness of our skin. All right, you see it's kind of in a brick and mortar structure, where the bricks are really mainly a protein environment, and the mortar surrounding or the barrier between the bricks is the phospholipid bilayers. And so really, if you think about this, inside of the bricks is more if it's protein soluble, water soluble, whereas the mortar is more lipid soluble. The intercellular route is the most common way when we have drugs that are well absorbed transdermally. This is typically the route that they take. On the upper left, you see, mechanical delivery methods. So there are, or recently there have been advancements in for example, vaccine delivery, where a matrix of very small microneedles mechanically disrupt this barrier. They poke through it to deliver a vaccine. You know, theoretically, you could use sandpaper and rub off a layer of skin until the stratum corneum is gone, and then apply the gel. But of course, that would be an awfully cruel thing to do. So the main thing is it's got to get through that. That's a difficult journey to make it through that. If you've seen 1883, as they're traversing the Oregon Trail, you got to be able to ride a horse, you got to be able to start a fire, fight off bandits. You have to have all these characteristics to make that difficult journey. Likewise, or to make an analogy, molecules need to have certain characteristics to make this journey through the stratum corneum. They have to be relatively small in terms of their molecular weight. They have to be relatively lipophilic, and this is actually kind of a Goldilocks principle. They have to be more soluble in oil than water, but if they're too soluble in oil, that doesn't work either. They have to be relatively potent. In other words, what is a typical effective milligram dose a day? As an example, fentanyl is very potent. It's measured in micrograms delivered per hour from the patch. I would never expect to see something like a metformin patch, where we need 1,000 milligrams or 500 milligrams per day of that drug. It's just simply not potent enough for enough of it to make it through the skin. It needs to have the right melting point. If it's too high, we're not going to be able. It will be a solid in the gel instead of a liquid, and then ionized or charged particles are also it's more difficult for them to cross barriers, and not going to get into the pKa's and Henderson-Hasselbalch equation, but there's a sweet spot with that one too. So ideally, our ideal molecule that's going to make this difficult journey possesses all of these or most of these properties. Commercially available dosage forms that are available transdermally, things like Butrans patch, clonidine patch, fentanyl patch, nicotine patch, transderm scope patch. If you're looking for a trivia question, that's the first transdermal dosage form that was FDA approved. Wanted to help you on Jeopardy later. All right, so those are the basics of transdermal absorption. Hopefully that makes sense. If you have a particular drug in mind where you're skeptical and you want me to send you specifics, I have that information. I think it's beyond the general scope of this, but I'm happy to send it to you in my emails at the end of this. I mentioned that I wanted to talk about interpreting studies that are out there, and key to doing this is understanding the difference between in vitro and in vivo, or one of the keys. In vitro is Latin, from Latin meaning in glass. So this is kind of where it's done in a laboratory setting. If you're to find something that says, a generic article title like new PLO gel improves absorption of haloperidol by 200%, it's almost a guarantee that they use an apparatus, something like this. And just to quickly explain it, there's this donor compartment at the top. They would be putting in their gel or solution there. Then there's this membrane right in the middle. They use for those membranes either human cadaver skin, animal skin, or different skin-like substances. There's a solution in the bottom and a spinner, and basically over time they draw from this sampling tube to see, like, okay, we put it in the top, and how much of it is in the bottom now? And they say, well, if we're measuring some in the bottom, then it's making it through the skin. It's making it through this membrane, okay? That's not the same as a live, intact human skin. So that's an important thing to keep in mind, but if you find anything that's supporting in the literature transdermal compounds, the vast majority of it comes from studies that use something like this that are not really applicable to live human beings from my perspective. I want to show you a couple of examples. I don't want to get into too much detail here, but there are different things that, just setting a drug on the skin and it passing through would be considered passive absorption. There are different ways to kind of hasten or improve that process. One of the ways is iontophoresis, where they add an electric current to it. And I just highlighted a couple of things. This is a in vitro, so in a lab study of chlorpromazine. They did a 5 milligram per milliliter, and after 10 hours, and this is in micrograms, they found 152 micrograms of chlorpromazine in that retrieval compartment. In the bottom with the iontophoresis, and I just highlighted 5 so we can do an apples to apples. They're giving it, the full current, the highest current on here, and we see, okay, wow, you got 1 milligram of chlorpromazine after applying this current. Of course, in hospice, we're not doing any of this. We're not applying an electric current, and even if we were, 1 milligram of chlorpromazine is far subtherapeutic, and it's really not surprising if you look at the characteristics of this molecule. It's too oil soluble, and it's not potent enough. In fact, it's even considered a low potency antipsychotic drug. Interestingly, and just continuing with the in vitro a group, and the reason I share this is that it's actually a study that was done in hospice patients, and they found that in vitro in their study, the PLO gels containing chlorpromazine they failed. They didn't achieve systemic levels, and not effective. Now on to the good stuff, and different from in vivo, what we're really looking for in terms of quality studies of this is in vivo, or in a live human being. Here we see chlorpromazine bioavailability from a topical gel formulation in volunteers, live human beings. They put on chlorpromazine gel, and I just kind of highlighted some of the pertinence. It wasn't detected in any blood samples. Now that's really important, because again, a drug has to get into systemic circulation before it's going to act on a receptor. And they measured at 1, 2, and 4 hours, and still undetectable levels of this. So if you're out there looking at research, these are probably 2 of the main things to look at. Is it in vitro or in vivo? And we want to be looking for studies that measure bioavailability via blood monitoring in live human beings when available. I'll say that, again, if you have a particular compound that you want me to share evidence with of why it's not a good idea to use it in a topical compound, I'm happy to do that. I think I have something for just about all the drugs that we'll mention today. And that's a good segue. Which ones should we avoid? Which ones are compounds that don't meet those 5 things and or have human studies that show that they don't get absorbed through the skin? I'm going to start with this one. Brad and I were chatting before we got going today, and this actually made a pretty big impact. 10, 15 years ago, the use of Ativan gel, Haldol gel, ABH gel, so they're throwing Benadryl in the mix, was pretty widespread. I think it was well intended, but it was pretty widespread. And our own main scientific body, American Academy of Hospice and Palliative Medicine, in a Choosing Wisely campaign, one of them was, don't use ABH gel for nausea. Don't use it. They stopped there, but I'll continue that. Don't use it for agitation. Don't use it for restlessness. Don't use it for anything. And the reason why is it is not absorbed through the skin. for the Ativan and for the haloperidol, they did find trace amounts of Benadryl, but not until several hours after putting it on. Now, that's another point that, I'm mentioning over and over again. Does it make it through the skin? But really, the vast majority of topical gel compounds, at least in my experience, are ordered on an as-needed basis. So when we order a drug as needed, one of the characteristics we're looking for is for it to work promptly. So even though the Benadryl, there was a little teeny bit of it, it wasn't showing up until, hours later and erratically. So this is kind of the first one to get called out because of negative evidence in live human trials. I'm going to add to that list, and I'm not going to get into specifics on these, but here are, from my experience, the top 15 different things that I see in topical compounds, topical compound gels and creams, where we should not be using them. And the reason that we shouldn't be using them is they don't possess the right characteristics, and or there are negative studies in human beings that show that it is not absorbed transdermally. And in things like, like clonidine, you might say, well, yeah, it's available in a clonidine patch. The main reason, I'll get into a study from the government on that one, but like some of these things, I think that's really the only one on here where it is absorbed, but it's just not, it's not a good item to include in topical compounds because it hasn't been shown to be more effective than placebo. So that's more of an efficacy one. All the other ones on here are really like a kind of just non-starters. They can't make the journey. They're not getting into systemic circulation and expecting an effect, and especially affecting a prompt effect from this is really, really wishful thinking. So I just want to share this story if you haven't heard this one, but this was kind of a scandal 5 years ago or so where there was rampant fraud and abuse in the military's program TRICARE, where they're paying hundreds of millions of dollars. At the end of this, they're spending $6 million a day on medications that contain ketamine, gabapentin, clonidine, lidocaine for these pain creams. Scientific literature in the Annals of Internal Medicine found that they're not better than placebo creams, and of course they cost a lot. If you're interested, the link's here, but you can see how this story ended. These folks, I don't know if they ultimately did, but they were looking at jail time for fraudulent prescribing of these. All right, that's the naughty list. Which ones are worth prescribing? And you'll see it's a shorter list. I'll start off with NSAIDs. There are things like ketoprofen gel where, based on the molecular characteristics and in limited literature available, it does. We do see local absorption with that one, but it begs the question, why use that one when we now have diclofenac gel that's generic, it's inexpensive. A good rule of thumb is to stick to commercially available products instead of compounds. NSAIDs, you can make a case that you can throw them into the mix in a compound, but generally speaking, we already have that. We have topical diclofenac. I'll mention lidocaine. Here, we see lidocaine patches, and the way I remember these is to keep it local. Okay, so if we're hoping for a local effect and not a systemic effect, it might be reasonable. So like lidocaine, those lidocaine patches work well for post-herpetic neuralgia, for other localized neuropathic pain. They're often included in miracle or magic mouthwash compounds where the patient can swish it around and temporarily numb, maybe painful mucositis. Another compound that's worth considering in select cases are opioid gels. These are almost always morphine and hydrogel. If we have inflamed open wounds and for whatever reason systemic opioids are not working well, we can definitely consider those. There's a good fast fact from the Medical College of Wisconsin on topical opioids if you're interested more. Another compound, topical, would be considered topical if you're spitting it back out, but we have these different rinses for mucositis containing things like ketamine, morphine, even doxepin sometimes, where it's rinsed and expectorated. That would be considered a topical local effect, and then every now and then we'll do some compound for bleeding management. But really, what you see in common with all of these is that they're all things where we're hoping for a local effect and not a systemic effect. And real quick, I think we're doing really well on time because half past. So let's get into the characteristic pushback that I've experienced in a decade plus of hospice consulting experience. And for me, it usually falls into one of two camps. And I was kind of thinking of these song lyrics as I was putting the presentation together. So Kurt Cobain, Nirvana, he's got a lyric, I know it's wrong, but what should I do? I think there's a lot of medical directors, prescribers out there that kind of feel held hostage in this. They know that, best case scenario, we're hoping for a placebo effect from these. They know that the drugs are not absorbed systemically. But for one reason or another, whether it's the group homeowner won't refer their patients to us anymore if we don't order what they want, or I know if I don't do it, the hospice across the street will do it, and they'll just go over and use them. Kind of a sad indictment of things that's the rationale for prescribing it. I hope eventually we move towards standing up for it, whether that's placing a call to those places, medical director to whoever's objecting for or advocating for using them despite the negative evidence will sometimes do the trick, or passing along supporting literature sometimes do the trick to make a non-believer. We have a believer and we want a non-believer with that. And then speaking of that, so that's one camp. The ones that they know it doesn't work well for whatever reason. Their arm has been twisted into prescribing it. And then the other camp, and a lot of this is nurses who are out there face to face with the patient. This is the Ron Burgundy, I don't believe you, of the group. I can tell them all day, there's a study that shows this, but they've been out there, they've put the ABH gel on, and then they saw her face, and now they're a believer. It's their strong perception that the compound has worked. And I just want to talk about that on the next slide here. Undoubtedly, many out there have put these gels on and the symptom improved. Okay, that those two things happen, but one doesn't necessarily follow the other. There's not a demonstrated cause and effect with those. So what else has happened while we're doing this? Well, number one, touch. Okay, gentle touch is being used to apply the compound. It's, at least in my experience, usually applied to the inner wrist, perhaps because our skin is thinner in that area. Presence, the clinician or the caregiver, whoever's putting this gel on is with the patient. They're being attentive with the patient. They're there. Okay, so they're with the patient to apply the compound. And then lastly, time. Okay, so after we put the compound on, time elapses, whether that's 10, 15, 30 minutes, an hour. for all we know, the symptom could have improved with time, with or without the compound. And based on what we know that the drug isn't getting into circulation, I would suspect that if we would have just put a placebo cream on, we would have seen the same results. There is a massive placebo effect in really any drug. That's why we do randomized control trials, placebo-controlled RCTs, so that we can account for this. And it's really doubly so with topical drugs. There's a strong mental tendency to, believe that when something is applied to the skin that it works. In fact, it was actually, for the diclofenac gel, relatively difficult to get FDA approval because they had to get bigger numbers to pull away from this placebo effect. It's so strong with topical compounds. So if you're one of the believers out there, if you came to the webinar today to see what's up, hopefully this resonates with you, that maybe it's not the gel, maybe it is the fact that I'm there gently touching the patient, being with them, time is going by. If nothing else, give it a try with something else, but ultimately, we owe it to our dying patients to give them a medication that we know is going to be absorbed and therefore has a chance to be effective. These topical drugs, topical compounds, going back to that slide with those 15 cards on it, we know that those are not good candidates for topicals. So we're doing them a bad disservice by not prescribing something else, whether that's in liquid forms of medication, commercially available topical forms of medication, parenteral medication, or rectal, again, with a suppository, or can't get it that way. Most drugs you can put through or use a Macy catheter to ensure that systemic absorption is had.